Our last group of organic molecules are called nucleic acids. Nucleic acids primarily have two functions. You may be familiar with the first. The first function is to store and transmit genetic information. There's also a nucleic acid that's very important that's involved in the energy transfer from one molecule to another. We're talking about DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. These are the two involved in storing and transmitting genetic information. And the third nucleic acid, ATP, or adenosine triphosphate, a very important energy molecule in your cells. So let's start by talking about what the monomer of a nucleic acid is. The monomer for nucleic acids are called nucleotides, and nucleotides all contain three parts. Take a look at this illustration. You'll see that every nucleotide has a base, a nitrogen base right there. Also has a phosphate group right here, that's a functional group, and a 5-carbon sugar that we're going to call a pentose sugar right there. And notice that the 5-carbon sugar is in the shape of a pentagon. There are 5 different nucleotides. They are adenine, guanine, cytosine, thymine, and uracil. We find 4 of these in DNA. The first 4, adenine, guanine, cytosine, and thymine. We find 4 of these also in RNA, adenine, guanine, cytosine, and uracil. Notice that thymine and uracil are in yellow. I've done that to emphasize that that DNA and RNA contain the same nucleotides except for those. In RNA, the thymine has been replaced with uracil. To form a polymer of nucleotides, we take the nucleotides. Let's look at this illustration in the bottom right. We are going to bond the nucleotides together to form a linear strand. Notice that the pentose sugar is in green, the nitrogen base here is in the yellow color, and you'll see the phosphate there to the side of the sugar. It's helpful to me when I'm trying to analyze a nucleic acid or figure out where my nucleotides are. The first thing I look for is the pentose sugar because it is in the shape of a pentagon. And I know that the base is going to be to the right and the phosphate group is going to be to the left. So if you'll notice in this illustration, I have nucleotides here bonded together. There's a nucleotide. There's the base, sugar, and phosphate. The phosphate of this nucleotide is going to be covalently bonded to the sugar of the next nucleotide right there. So there's a nucleotide. That phosphate is bonded to the sugar of the next nucleotide. So this is a linear strand of nucleotides covalently bonded together between the sugars and the phosphates. We call this bonding, the sugars and the phosphates, we actually call that the backbone of the molecule. Those sugar-phosphate bonds are the backbone. So if we were going to take a look at RNA, RNA is a single-stranded molecule of nucleotides that are covalently bonded together. That's basically the structure we just analyzed. Here's my RNA right here. Notice that's uracil, adenine, guanine, and cytosine right there. So let's take RNA and take a look at it molecularly. Let's break it down. Here's my sugar of one nucleotide. There's the base and the phosphate. So there's a nucleotide. That phosphate's bonded to the sugar of the next nucleotide. So you can see that this is a single strand, a linear arrangement of nucleotides covalently bonded together. Well, DNA is double-stranded, so it's actually 2 of those strands bonded together. So let's take a look at how that would work. First of all, you recognize DNA as this structure. This is what you're most familiar with when you think of DNA. You think of it as a double helix type structure, kind of like a slinky. Or if you want to think about this as a spiral staircase, We could actually look at it in terms of a spiral staircase. This would be the handrails of the staircase, the gray portion, and the colored portions in the middle would be the steps of the staircase. So we're going to take this staircase that you see right here, and we're going to unwind it so that it lays flat like a ladder. And let's look at it and talk about it like that. So if this were my ladder, if this were my spiral staircase that I laid flat, these blue and yellow structures right here, those would be my handrails. Here's my other handrail right there, and this would be the steps. Okay, well, this is actually a strand, and this is a strand of nucleotides bonded together. So let's cut it down the middle. The broken dotted lines right here represent hydrogen bonds, and you see that the bases, adenine and thymine, here are bonded together right there with a hydrogen bond. Here's cytosine and guanine, there's a hydrogen bond. So let's just split these down the middle and talk about one strand right here. If we take this strand, you can see that this is nucleotides covalently bonded together. Here's my adenine, there's my sugar, and there's my base, so that's a nucleotide. Here's my sugar, cytosine and phosphate. There's the second nucleotide. And I can go on up the strand, and you can see that that's a linear arrangement of nucleotides that are bonded together. So this is also a strand. It kind of looks different, and that confuses students sometimes because it's oriented in a different direction. Take a look at the sugars. If you're not understanding what I'm meaning, take a look at the sugars. These sugars are pointing upward, and these sugars are pointing downward. So they're actually anti-parallel or oriented in opposite directions. So the two strands are held together by hydrogen bonds, and then they're twisted to form a double helix there. Also notice that our adenines are always bonded to thymines, and our cytosines and guanines are bonded together. This is what is called complementary base pairing. A goes with T and C goes with G. Okay? So our special nucleotide, our third, is called ATP, or adenosine triphosphate. It is an adenine, a ribose, and 3 phosphates. That's why it's called triphosphate. Tri means 3. So we have our adenine here. We have our 5-carbon sugar right here. That's a ribose. And here is my 1, 2, 3 phosphates right there. Okay, so this is a very, very high energy molecule. As a matter of fact, we're going to call this molecule the energy currency of the cell, because the cells are going to actually spend the energy in this molecule. We learned in chapter 2 that energy is stored in bonds. So very important bond is this bond right here, that 3rd phosphate right there, that bond. If I were to break that bond right there, approximately 7.33 kilocalories of energy is given off. And that's exactly the amount of energy that your cells need to function. Anytime your cells need energy, they actually break the bond to that 3rd phosphate bond and use the energy from the breaking of that bond. When we break that bond, it's called hydrolysis. And when that bond is broken, think about it for a moment. If I break that bond, I take a phosphate off, I actually have adenine, ribose, and 2 phosphates left. Well, a prefix for 2 is di. So if I break that bond or take that phosphate off, I have adenosine diphosphate, adenine, ribose, and 2 phosphates. I actually have a loose phosphate, and energy has been given off. So very important reaction. When your cells need energy, that's where they get it. So here is an illustration. Here's my ATP right there. I'm going to break that bond, that 3rd phosphate bond. Energy is released, and I end up with ADP and P phosphate. So some of you may be wondering, what happens to ADP and P? Where does that go? Most of you learned somewhere in high school biology that the mitochondria of the cell is referred to as the powerhouse of the cell. And that's because the ADP and the P are actually going to go into the mitochondria. The mitochondria takes the energy from the food that you eat, takes that energy to bond that phosphate back onto ADP to form ATP. So your mitochondria is responsible for the production of ATP that your cells are actually going to use.